The next session, our speaker for today is Dr. Hemlata Ganeshan, Professor, Every College of Applied Science, Oman. It's our pleasure to introduce Dr. Hemlata Ganeshan, a highly accomplished academic and researcher with over 18 years of teaching and research experience. Dr. Hemlata completed her PhD in Information and Communication Engineering from Anna University, Chennai in 2014. She is currently serving as a professor IT department at the Ibri College of Applied Science, Oman, where she imparts her expertise to both undergraduate and postgraduate students on diverse subjects such as database programming, data mining, and artificial intelligence. Dr. Hemlata is an Oracle Certificate Associate and Oracle Certified Professional and has successfully trained more than 250 students to achieve these globally recognized certifications. She is also an industrial certified AI engineer. Her research contributions are noteworthy with more than 10 papers published in reputed journals, 2 book chapters, and 10 conference papers. She has successfully secured and executed 2 funding research projects and serves as a reviewer for 4 esteemed journals, underlying her dedication to academic excellence and scholarly rigor. Furthermore, she has previously served as a trainer for industry-driven programs such as Infosys Campus Connect program and Wipro Jumpstart program, bridging the gap between academia and industry. Her active involvement in curriculum development and industry outreach initiative further highlights her versatile contribution to academia and beyond. Dr. Hemlata Ganeshan is a valuable resource for this faculty development program, and we are honored to have her with us today. Ma'am, welcome, ma'am.
Yeah, thank you, ma'am. Thank you for your warm welcome.
Thank you, ma'am. You can start your session, ma'am.
Yeah, I will just share my screen.
Sure, ma'am.
Can you see my screen, ma'am?
Now only it's yes, ma'am. It's visible, ma'am. You can start.
Okay, fine. Okay, good evening, one and all. I welcome you for today's session. I'm very happy to share a session in this ATAL workshop. Today, my topic will be about genomic sequence analysis using large language models. So before I get into this topic, I just want to narrate what we will be discussing today. We'll be discussing about genomic analysis. So I just want to cover in a few slides what is the importance of genomic analysis. And I'm going to drive you to the path how artificial intelligence can be used in genomic analysis. And then later on, I will be covering about what is LLM models and what are the LLM models that are used in this bioinformatics. So we are going to discuss about some pre-trained LLM models. Then I'm going to show you practically how to implement these LLM models in your own data set. Okay, so this is what we are going to cover in today's session. So let's get into the details. So what is genome? Genome is either a DNA or RNA is called as a genome. They are nucleic acids. So you know any form of life in this earth has a DNA. Yes, sir?
I'm putting slides on.
So you cannot see the slides?
Full screen mode now.
Well, it is full screen only for me. Let me share again. Can you see, sir?
Yes,
Is it full?
no.
Okay, thank you. So any form of life in the earth has a RNA or a DNA. These are called as genomes. Okay. So, and a genome can be either a unique RNA or it can be a DNA. So, if you take any virus, any bacteria, or any fungi, or a plant, or a human being, okay, you take any form of life in this earth will have a DNA, either DNA or it will have a RNA. So, DNA is called as deoxyribonucleic acid, and RNA is called as riboxynucleic acid. And either DNA or RNA, it consists of only 4 nitrogen bases. For example, you take a DNA, it has 4 nitrogen bases, which is called as adenine, thymine, guanine, and cytosine. So this is the combination of DNA. And if you take a RNA, it has only one change. The T in DNA is replaced by U in RNA. So this is how a genome will look like. So a genome can be either RNA. So you can see RNA is single stranded and DNA is double stranded. So it will be like this. Okay. So this is a genome sequence. Now, for example, you take a fruit fly. Okay. It has genes of around 13,600 base pairs. What is this base pairs? These are only called as base pairs. So you can see this adenine, thymine, cytosine, guanine, they'll be keep on repeating. So here you can see it's keep on repeating. Okay, each one is called as a base pair. So how many base pairs are there in a fruit fly? You have around 13,600 base pairs. Okay, in a tapeworm, you see around 19,500 base pairs. In a human, not very special when compared to a tapeworm, you have only 20,000 to 25,000 base pairs. Rice, you have 45,000. Maize, you have 50,000. Okay, so yeah, this many base pairs are available. And you know one thing, still they are not able to find out the entire genomic sequence of humans. Okay, I think last 2 months before, the scientists were able to discover the entire sequence of a human genome. Okay, that much complex it is to discover the genome sequence. Now, what is the process in extracting the genome sequence? You take an organism. Okay, so from the organism, you try to extract the DNA sequences. For example, if it is a human, they'll take a blood from us, and from the blood, they will extract the DNA from the blood. And this DNA, it will be fragmented into 4 small pieces like this. Okay, and it will be given to a sequencer. You have a machine called a sequencer. So this DNA samples will be fed. Okay, the fragments will be fed one by one to the sequencer. The sequencer will give you the structure of the genome. Okay, so this is how it will be done. Now, this process itself will take a long time. Maybe depending on the complexity of the DNA, maybe it will take 1 week or 2-3 days at least to get the sequence. Okay, so a lot of researches are going on in this field, how to improve the speed of sequencing. Okay, because if any new virus or any new disease comes, you should be very quick in diagnosing it. As we should find out what type of virus it is. So the first step we need is sequencing. So a lot of researchers are going on in this field to speed up this process. Okay, this is the first step. So after you get the DNA sequence, now you have to compare it with your known database. So we have a known database. For example, we have NCBI. Okay, so here we have the DNA samples of many species, many animals, humans, okay, viruses, bacteria. Okay, many species DNA samples are stored in this database. So once you get your DNA sample, you can compare it with a known database to find out what type of virus it is, whether it is a new pathogen or it is a mutated pathogen or whether it's already existing pathogen. So you have to find out by comparing with the database. So that is another area of research, how to efficiently classify the sequence. That is another area of research. So quickly it has to classify because your database is very large in size. Okay. For example, it is in trillion and non-union size. Okay. That much big amount of database we have regarding the viruses and bacteria. So you have to compare with the database and quickly find out to which sample it is matching. Okay. That is another area of research. Okay, so another area where it is used is nowadays we have some genetic disorders. We frequently hear, right, some genetic disorders in children's. Okay, so when there is a growth disorder, okay, or the development, its process is delayed, then we suspect there is a genetic disorder in the children. So what we do, we take blood samples from the children, we do the process of sequencing. So sequencing is splitting the DNA into fragments like this, and then feeding the fragments into the DNA sequencer machine to get the DNA sequence. And then after you get the DNA sequence of the patient, you find out to which disease it matches, okay, which pathogen are detected in the patient genome to identify whether he has a genetic disorder. This is also not very easy. It takes long time to do this deduction. So researchers are again going on how to make this process quickly. So in all these fields, you can see artificial intelligence is used to make this process much quicker, so that the disease can be analyzed much faster. So the role of genomics in healthcare is enormous. So genomic analysis is used for diagnosis, it is used for personalized medicine, it is used for preventive care, and is also used for drug discovery, etc. and etc. So we feel now genome plays a vital role in most of the disease prediction system. Okay, so what we are going to talk in this session. In this session, so as I told you, there are a lot of researches are going on in the field of genomics. What I am going to concentrate, I am just going to concentrate only on classification. Okay, so for example, when we got the pandemic, right? So when the patient was affected by some viruses, so the first thing we have to do, we have to identify what type of virus it is. So we have to collect the DNA or RNA from the affected patients, and you have to check your database, okay, whether it is a new virus or whether it is a mutated virus or it is already existing virus. So as I told you, we have 10 nonillion individual viruses exist on the earth. So the database is such a big one. Okay, so what you have to do, you should be able to identify the taxonomy classification of the virus. Okay, then only you can deal your pandemic in a better way. So the first step is identifying the taxonomy. So it's nothing but classifying the virus, okay, or classifying the DNA or the RNA. It can be a DNA or RNA or whatever it may be. So the first step is required. Either it is a disease or whether it is an outbreak or whatever it is. The first step is we have to identify to which family it belongs to. Okay, then only we can take the next step. So how to do that classification very effectively? It takes a long time to do classification. So how to do that classification effectively? That is what we are going to discuss in this particular session. Okay, now, so this is how you have the database. Now this one, I have taken it from NCBI. So this is the database where you have the sequence, the DNA sequence and RNA sequence of all the viruses, bacteria, plant, human, all type is available in this NCBI. So here you can see, for example, let's take coronavirus. The virus got mutated and you have different samples available in different part of the world. All the samples are fed into this database. So it is available in this database. So if you want to extract the DNA samples for these viruses, for example, we had MERS, we had corona, then we had SARS, right? We had Deepa virus, right? We have a lot of viruses, right? So if you want to get the DNA samples of all these viruses, yes, you can download it from NCBI, and the file is available as a FASTA file. Okay, so here you can see the session number for that one and the sequence that is collected and for what type of virus, okay, from which country it is collected. All the details will be available in this NCBI. So from that you can download and get the samples of all the viruses. What are the samples you need? Okay. So now, after getting the DNA samples, now what is our aim? Our aim is to do classification effectively. So we should identify, if I give a DNA, you should tell me whether it is a DNA of a COVID, or it is a DNA of a MERS, or it is a DNA of a SARS, or what it is, okay, or hepatitis B, or whatever it is. So if I give a DNA, you should be able to identify the label, or you should be able to identify the class. So that is our objective. Okay, so the first and foremost thing that we have to do, we have to apply, so what are the standard techniques available? Either you can apply machine learning or you can apply deep learning. Okay, so whatever it may be, you can see the DNA samples are like English like language, right? Because it has alphabets, right? So machine learning cannot process such categorical data, right? It has to be numerical data. So the first and the foremost step that is required is we have to convert this one into numerical values. So to convert this into numerical values, there are different ways are there. Either you can follow the nominal method or you can follow the ordinal method. In nominal method, we have one hot encoding, okay, or you can go to ordinal method where we have label encoding. I'm just discussing the basic methods. So if you take a raw DNA sequence, so this is your DNA sequence, we are going to convert that into numbers or call vectors, okay. So we will follow one hot encoding scheme. So what are the basic components we will see? So the N is not clear, okay. So as I told you, a human genomic sequence has around 20,000 base pairs. Okay, in some parts there is a gap. That's why the researchers are not able to find out the complete structure of the human genome sequence. They're not able to find out because there are some gaps between. So the gaps are represented by some value called as N here. Okay, so you have A, C, G, T are the basic pairs, right? N is unknown. Now let's take the first one. So first one is T, right? So wherever you have T, there you will put 1, all the others will be 0. The next one is A. So wherever you have A, you will put 1, all the others are 0. So like this, you're trying to convert this DNA sequence into a vector, okay, into numbers. So this is what is required. So every sample will be converted into a vector representation. Okay, that is your one hot encoding. The next one is label encoder, where each one is given a unique value. Okay, for example, A will be represented as 1. Okay, C will be represented as 2, G will be represented as 3, and T will be represented as 4. Okay, so like this, you assume values for each one and convert your DNA sample into numbers. Okay, or there is another method that is called as k-mers. Okay, so if this is your DNA sample, you have to generate k-mers. So what is this k? K can be any size. It can be 2, 3, 4, 5, 6. It can be any. Okay, so I will take the first 6 digits. Okay, the next I will take next 6 digits. Okay, from starting from here. Then I'll take next 6 digits. So it is overlapping. So this is called as overlapping k-mers. You also have non-overlapping k-mers. So there are different types are there. So you can run your algorithm for each size of k and also for each type of k-mers. One is okay, one is non-overlapping and overlapping. So you can run your algorithm for different values of k-mers and then check which one is better. Okay, so like this, your DNA samples has to be converted into numbers. Now, after converting into k-mers, now we will use bag of word model and convert that into numbers. Okay, these are nothing but frequencies, how many times it appears. Okay, each one. GTCA, how many times it appears. So here you can see GTCA, it appears 2 times. So this is called as what? Bag of word model. Okay, so this bag of word model is nothing but the frequency, how many times this pair appears. Okay, the frequency. So now we have the DNA sample. So this is your DNA sample. Now DNA sample is converted into a vector, okay, into numbers. Now after you convert your DNA samples into numbers, now you can use either machine learning algorithm or you can use a deep learning algorithm to do the classification process. So now I just discussed only the 3 methods, right? Just I discussed one hot encoding, I discussed label encoding, then I discussed k-mer counting. Okay, k-mer counting is done with bag of word model. Okay, there are many. Okay, so bag of word itself you can do with unigram, bigram, trigram, and n-gram. Okay, so bag of word model. So here I told you k-mer size is 6, right? So you can start with size 1 unigram, size bigram size 2, okay, trigram size 3, and to n. So you can repeat many things and you will use bag of word model. Either you can do that or you can use this method, term frequency, inverse document frequency. This method can be used to convert your DNA samples into numbers. Okay, or you can use word to vec method. This is more efficient than all these methods because these methods just take the frequency into account. Okay, it does not take any meaning for the words, right? Okay, so it just takes frequency to accounts. But word to vec method is a little different. It converts word to a vector and the word will be mapped based on the meaning of the word. Okay, so word to vec method can be used. Now after you convert DNA sample into number, as I told you, so here you have DNA sequence, you're converting DNA sequence into number. Either you use one hot encoding or label encoding or bag of word model and convert that into number. After converting that into number, Now you want to divide your data set into train and test, and then you can use any of the available machine learning algorithm and do the process of classification. So very simple. So here the work that you need to put is only in pre-processing the DNA sample. So you have to extract from NCBI, you have to do the pre-processing step, you have to convert your DNA samples into numbers, then easily you can apply all the machine learning algorithm and do the classification. So this method is like a state-of-art method. It's already available, okay? Now, you can also create a deep learning method. Okay, for example, here you have the DNA sequences. Okay, convert the DNA sequence into numbers, and then you can create a deep learning model. So here the deep learning model is, here you have to use one-dimensional convolutional layers, not two-dimensional. Two-dimensional is mainly used for text and videos. Okay, so this is only numbers, right? So you have to use one-dimensional convolution model. Okay, CNN, one-dimensional CNN model, and then you can do the classification. Okay, or you can also use LSTM architecture. So these are all already existing, right? So LSTM architecture mainly is used for a sequence. The DNA is also a sequence. So LSTM performs very well if it is a sequence. Okay, so for example, you want to predict the next sequence. Okay, so it can perform very well if you use LSTM. So here you can also use LSTM. So the only thing that is necessary is you need some numbers. Okay, your DNA or RNA should be encoded as numbers or vectors, okay, encoded as numbers. And then you have to give it as an input to the convolution layer. On the convolution layer, you will also add an LSTM layer. And then finally, you will do the classification. Okay, so these are the three methods which are already existing. So still now, I just discussed what are the existing methods available. Okay, so just to recollect what we learned still now, just we are going to convert our DNA samples into numbers, and then we are going to classify using machine learning or deep learning, or we are going to use LSTM network to classify the sequences. Okay. Now, what is the main topic of our session today? Our session today, we are going to cover how to use large language models to do the DNA sequence classification. Okay, that is what is the main topic today. So before we are going to see the models that we are going to use, we should know what is LLM. Okay, what is large language models and what is the basic architecture of this LLM model. Then only we can understand how it is better than the existing methods. So I told you three existing methods. Okay, machine learning, deep learning, and then I told you LSTM. So how these LLMs are better than that, that is what we are going to discuss in the upcoming session. Okay, so before I get into LLM, nowadays you frequently hear the term LLMs everywhere. Okay, from where you hear this term? Because of ChatGPT, right? So what is the history of this language model? Okay, so in 2016, they found out transformer. Okay, this only created a major change in the field of NLP, that is natural language processing. This transformer created a great change in the NLP. With the help of this transformer, they created a first large language model, which is called as BERT. And later you can see you got GPT. And now you have GPT-4, BARD, GPT-4 model. You know how it is, right? So mostly they are used for many natural language tasks. See, so this one is before transformer. This one is after transformer. So after inventing the transformer, you can see there is a lot of enhancements going in the field of NLP, right? So what are the applications of transformers? It can be used for text classification. It can be used for text summarization. Now, if you want to write a letter, you go to the chat GPT, you ask a question, it gives you an answer, right? So you chat as if you are chatting with a human, right? So it's able to generate text. It is able to identify. It is able to classify, right? So it is very powerful because it uses the architecture of what? Transformers. Okay, so it is able to recognize the entity. So if I give a name, it's able to recognize whether it is a person or place. It's able to recognize text generation. Okay, translation. It converts from any language to any language. Language modeling, question answering. So these are all the applications of transformer. So okay, so what is the special thing that is added in the transformer, which made the transformer more powerful? That is what we are going to discuss. See, before, all the words were converted into vectors, right? You did not understand the word. Just you took a word and converted that word to a number, okay, either 1 or 0. You did not consider the context of the word. Okay, the context of the word was not considered. But after creating transformers, so before transformers, you see, transformer was around some 2016, right? So before transformer, they did something called as what? Attention mechanism. So it means it started understanding the context of the word. The word context was able to understand. So that is called as what? Attention mechanism. So this attention mechanism only is used in the transformer. That only made transformer more powerful. So it is able to understand whatever you speak, the transformer is able to understand because it understands the context of the word, how you speak, okay? So it sees what is the word before and it also sees what is the word after. So it does not see only the neighbor words. It is able to it is able to travel through a long sentence. See, in LSTM is also sequence, right? So it will just see only the neighboring word. Okay, LSTM will just see only the neighboring word, but transformers are not like that. It's like humans, okay? So it will able to go through a long sequence, able to remember the sequence for a long time. Suppose, for example, I'm asking you, hi, how are you? Yesterday you went to hospital. How is your health now? So it remembers the sequence, okay, and it generates the word more efficiently based on the sequence. Suppose if you don't remember the sequence, then the result that is produced by the transformer will not be good, right? So the special thing that is used in the transformer is called as what? Attention mechanism. Okay, so based on this only, many large language models are developed. Okay, now before this attention mechanism, I told word to vec was used, right? So for example, here I told you word to vec, right? Yeah, here I told you word to vec. So this also will convert a DNA sample into number. Any word you give, it will be converted into number. So that was an efficient technique. For example, if you see, it will plot the word in a three-dimensional space. Okay, word to vec will plot the word into three-dimensional space. So if you take man, it will give you a vector. Okay, for man there will be a vector. For woman there will be a vector, different vector space. Okay, king, you will have a different vector space. Queen will have a different vector space. So it is able to differentiate man and queen. It's able to differentiate king and woman. So each one will have a unique vector representation here. Okay, so each of these words will have a unique vector representation because it's plotting the words in a three-dimensional space. Okay, for example, see, walked, walking. So this will have a unique vector representation. This will have a unique vector representation. Okay, so word to vec was efficient. For example, see, the country and capital, even these words were having unique representation. So word to vec was efficient. But when it comes to like this, see here, we went to the river bank. I needed to go to a bank to get a deposit. So here also you have the word bank. Here also you have the word bank. Okay, but this bank is different. This bank is different. So if you give this sentence to word to vec, okay, word to vec. If you give this sentence to word to vec, this bank and this bank will have same vector representation. Okay, why? Because it doesn't take the context into consideration. So when you take the context, this is talking about river bank. This is talking about the bank because you're going to make a deposit. So this bank and this bank is different. Okay, so word to vec will not take the context into consideration, but your attention mechanism will take the context of the word into consideration. Okay, so based on that concept, we created an architecture for the transformer. So this is the architecture diagram of the transformer. Okay, now this architecture diagram we should understand because this architecture only is used in all the large language models. Okay, so for example, here what will happen? You are giving an input. Suppose, for example, you are doing a chat with chat GPT. So what you do, you are asking it to generate, you are asking it to write a story. Okay, give me a, you are asking the chat GPT to generate a story. So how it will generate a story? The chat GPT is being trained with the entire corpus. Okay, entire internet. Okay, all the it is trained with all the entire internet. Okay, so when you give a question, so it is try to predict the first word. So how it will try to predict the first word? What is the question you ask? The question you asked is, give me a story. That is a question. So it is already trained with the internet, right? So it tries to predict the first word. So what will be the first word if you have a story? Mostly it begins with what? Once upon a time, right? So the first word will be once. Okay, now that will be added to the answer. Now, if the first word is once, what will be the next word? Once, mostly it will come with what? Once upon a time, right? So upon a time will be added. So like that, it starts generating words one by one, and then it gives you the answer as a whole. So this is the concept that is used in the transformer. So for example, here you see we give an input to the transformer. So first our input will be divided into tokens. Okay, this is the first step. And then our tokens will be converted into embeddings. Embeddings means it will be converted into numbers, vectors. Okay, and then we also should consider the position. Why? Because here you see, based on the position, here the word bank has a different meaning. Okay, when it comes along with the river, the meaning is different. When it comes along with the deposit, it comes with a different. So position is very important to understand the context. So for every token, position is also added to that token. And then it is fed into a series of transformer blocks. Not one block, see, series of transformer blocks. And then the last layer will be your hidden layer. Okay, so you will get the output from the hidden layer and then give it to a softmax function. So softmax function will convert the vector to a probability. So which word is having the higher probability? That word will be your next sentence. So here, see, write a story is your first, is your input. Okay, the first word that we get from our transformer is once. Then that will be added to the input. What will be the next word? So like this, slowly it starts generating. Okay, generating the entire story will be generated because the network is already trained. Okay, so the first step, as I told you, is the first step is called as the tokenization. So here you give a sentence and it's divided into tokens. So here you can see it's divided into tokens. And each token, you take each token will be converted into a number. Okay, so this is vector. Okay, this is called as embedding. Okay, for example, write will be converted into a vector. A will be converted into a vector. Story will be converted into a vector. So like that, each token will be converted into a vector. Then to that one, you're going to add the position because position is very important to understand the context of the word. So you add the position. So write is in first position, A is in second position, story is in third position, dot is in fourth position. So like that, you add the positional encoding. And then you give your input to the sequence of transformer blocks. So why sequence of transformer blocks? See, the first thing you have in the transformer blocks, you have attention mechanism. So attention mechanism is like, for example, I am giving you a text. Okay, I'm asking you to read the text. First time you read, you will not understand much better. You read it again. Okay, second time you read, you will understand much better. Then you read it again. So when you read it many times, you get better understanding, right? So that is why you have different blocks of transformer being repeated. Okay, so you have series of transformer blocks. So your input passes through series of transformer blocks so that your transformer clearly understands the context of your input. Okay, so that's why it understands everything. And it also speaks like a human. How human understands, the same way the transformer also understands whatever we speak, right? So that is because we have attention mechanism and not one transformer blocks. You have series of transformer blocks. Okay, so this is what is your transformer architecture. So when it passes to the attention mechanism, it reads this one. Okay, money in the bank. So now it understands. For first time when it reads, it will not understand. So when keep on reading, then it will understand, okay, money is an important word in this sentence. So when money and bank comes together, then understand it is the, so first it will have a confusion whether it's a river bank or money or the bank. So after you pass it through the attention mechanism, it will be clear that this bank refers to the organization where you deposit money. Okay, so this is what is the attention mechanism. So transformer is using that attention mechanism. So you give your input to the transformer, and the last layer will be your softmax layer. It will convert the vector into probabilities. So you can see for every word you have a probability, right? For every word you have a probability. Now, which word has more probability? Once has a higher probability. So what will be the next word? When you give write a story, what will be the next word you will get? The next word you will get is once. Okay, once upon a time. So you will get the next word as once. So this is the concept of transformer. Okay. So based on the concept of transformer, they created a first pre-trained model, and the model name is called as BERT. Okay, so what is BERT? So maybe you would have heard this model frequently. What is this BERT? BERT is bidirectional encoder representation for transformer. Why it is bidirectional? It means when it's try to understand a sentence, it don't scan through the left side or right side. It uses both the sides. Okay, so it keep reading both the sides, to the left as well as to the right. That's why it is called as bidirectional. And actually, a transformer has two things: encoder and decoder. Okay, we are not going to discuss decoder because all our pre-trained models are only based on encoders. So I'm not discussing the decoders. So actually, you have decoders also. Okay, but we are going to consider only encoder for all the models. So I just stopped with encoder. So it is bidirectional encoder representation for transformer. This is the BERT. Now, what is the architecture of the BERT? This is the architecture of the BERT. Okay. So, so all your language models, see, they are using the transformer model only. And you see how many blocks of transformer model here you have. 12 times the transformer block is repeated. Okay, so 12 transformer blocks are there. Okay, so what will be your input? Your input is write a story, right? So anything can be your input. So inputs are tokens. So your input has to be converted into tokens. And then what concept BERT actually uses to learn is, see, BERT is a pre-trained model. This model is already existing. They already trained this model on the entire corpus. Okay, so the model is already trained. Okay, but if you want to use your BERT for your own data, for example, I want to use it for our college purpose, then I want to train on my data, right? So because it's already trained, okay, it is used for natural language processing. So you want to do text classification, you want to do summarization, you want to do anything, then you will be using BERT, right? So you can use BERT, but on my own data set I want to do, then I have to give my own data set as an input. So how BERT will start learning your data set? Actually, it uses two techniques to learn our own data set. Okay, we can use our own data set. So for example, now we have chat GPT, right? So many companies, what they did, they customized chat GPT for their own purpose. Okay, so I can customize chat GPT and use it for my college purpose. So maybe students can answer what course I can take next semester to the chat GPT. Because I'm not going to use this chat GPT as it is. It's already trained. It's a pre-trained model. I'm just going to use that chat GPT on my data set. So I'm going to give my own data set to the chat GPT. For example, the courses that is offered in our college, what are the which teacher is teaching which course, what is the credits of the course. So like that, I'm going to give some more data to my chat GPT, and I can retrain my chat GPT. Okay, and then I can customize it and use it for my own purpose. That can be done. Similarly, BERT also. So BERT is already pre-trained on a large corpus. Okay, now we have to use a BERT for either text classification or translation or summarization or next sequence generation. For any purpose, you can use your BERT. Okay, so how BERT learns from your data set? That okay, so it uses two techniques to learn. Okay, one is it uses masked language model and it uses next sentence prediction model. These two models are used by the BERT to understand the data set that we give. Now, if you take this BERT, you can see there are 12 transformer models are repeated. Okay, and then you can see inside here one small change when compared to the transformer model. Here you have multi-head attention. Okay, so in transformer model, if you see previously the architecture of transformer, you have only attention here, right? Attention, and you have a feed forward network. But here a small change in BERT. Here you have multi-head attention and a normalization layer on top of that, and then you have a feed forward layer and then a normalization extra. Okay, only a small change is done to the transformer model in the BERT. Okay, what is this multi-head attention? For example, if you are diagnosed with a disease, what you do, you will take a second opinion from another doctor. Maybe that doctor is a specialized doctor, and you may take another opinion from another doctor from another specialization. Okay, so it's multi-head attention is like that. Okay, you read a sentence, okay, you read it again, okay, because the transformer blocks are repeating, right? You read it again, until you understand the context of the sentence very clearly. So how many times you will be reading? You'll be reading 12 times because the transformer blocks are repeated 12 times, and each one is an attention. Attention means what? It takes the context of the word clearly. Okay, so here multi-head attention. So it means in different perspective you read the same word. Okay, so that you better understand the word. Okay, that is the difference. So this is a small difference that is added in the BERT model, but basically it follows the transformer model, and it has only the encoder in it. No decoder is there in the BERT. Okay, so as I told you, how it learns from our data set. When I give my data set, okay, I want my BERT to understand much better. So what I will do, I will mask some tokens, and I will and my what my BERT will do, my BERT will try to predict the masked tokens. By predicting the masked tokens, it will try to learn my data set. Okay, so for example here, so as I told you, we have masked language model. So this is how the training is done to the BERT. So I will repeat again. See, BERT is already trained on the entire corpus. Okay, now I want to use BERT for my data set. What is my data set? For example, let's take my data set as a DNA data set. So it has to learn my DNA data set properly, right? So how it learns my DNA data set, when I give my DNA data set as an input, it converts my DNA data set into tokens, and it will mask some tokens. Okay, mask means it will hide some tokens. Okay, and it will try to predict that token. Okay, and then it checks whether its predictions is correct. Okay, so if the predictions is correct, it will start learning our data set. Okay, and then we can use our BERT for classification or text generation for any purpose. That's how the birds are trained on the new databases. Okay, so that is your masked language model. It also uses new sentence prediction model to train. Okay, let's see what is this masked language model. As I already told you, if this is a sentence, okay, this sentence is there in the data set. Okay, the man went to the store to buy the milk. So it is there in the stores. Now I want my BERT to understand the meaning of this sentence. So how it will understand the meaning? It will divide this into tokens. Okay, for example, let's take it divides this into tokens. After dividing it to token, it will mask some tokens. Okay, randomly it will mask some tokens. Maybe recommended is 15% of mask should be there. Okay, so it knows the answer. Yeah, it has the answer. Okay, but it will try to mask some random tokens, and it will try to predict that tokens. Okay, so it will try to predict. A man went to the stores to buy gallons of milk. So it will try to predict. So if the predictions are correct, then it is trained well. Do you understand? So it keeps on training like this. So like that, slowly it starts learning the new data set. Okay, so you should not mask too much. So if you mask too much, then you will not have enough contents to learn. So BERT will not learn properly. If you mask too little, then it will be very expensive to train because you will mask only too little. So it should be a optimal value. Tokens has to be masked. Okay. The next type of method that is used to train the BERT is next sentence prediction. So it uses this also. See why they use all these things? To learn the new data set. They are learning. Okay, learning is not very easy, right? It's like making the child to by heart something. Okay, so you are teaching the child. For example, you are teaching the child the photosynthesis. So how will you teach? You will teach the full scenario, right? One by one, you will teach the scenario. Then you will ask some questions to the child. Okay, now tell me what happens when the sunlight falls on the leaves. You ask some questions to the child, and you check whether the child answers correctly. The same concept. Okay, the same concept is also used in the BERT model. Okay, so what you do, you take two sentences and you check whether these two, when this sentence comes, this is the next sentence. Okay, so it's like a binary classification. So you give two sentences, it will classify is next sentence or not a next sentence. For example, these two are not next sentence. Man went to the store, penguins are flightless. These two sentences are not next sentence, but these two are next sentence. So it also use this method. Okay, why they use all this method? So that BERT understands much better about the data set. Okay, so now in BERT itself, you have different models. You have a base model and you have a large model. Okay, So this is your transformer model. So in transformer model, we use only 6 layers of transformers. In BERT, you use 12. In base model, you use 12 layers. Okay, in large model, you will use 24 layers. Okay, and this is about the hidden layers. Okay, the last layer that you add to the BERT will be your hidden layers. Okay, hidden layers. So how many neurons you have in the hidden layers? So here you have 768 neurons. Here you have 124 neurons. In transformer, you have 512 neurons. So what is this actually? See, you are giving a sentence. For example, hi, okay, how are you? Okay, so this is your sentence. Now this will be divided into tokens. This will be divided into tokens. Now this token will be converted into a vector, right? Token will be converted into a vector. And then positional encoding will be added. Okay, then it passes through the entire transformer. And when it comes to the last layer, that is the hidden layer, how many neurons are there? You have 768 neurons are there in the hidden layer. So each token size will be 768. Okay, the vector size will be 768. Okay, I will show you when I show you the Python code for this one. So each one will be converted into a vector of size 768. Each token will be converted into a vector of size 768. Okay, so these are the different BERT model we have. Okay, so you can use this BERT model for any NLP task. Okay, now as I told you, you can use it for classification, you can use it for translation, you can use it for question answering. Okay, many tasks you can use the BERT. Okay, now coming to the main part. So still now I were talking about LLMs in natural language processing. Can I use these large language models for biomedical field? Yes, these large language models can be used for biomedical field because you can use it for even DNA sequence classification, because DNA is very similar to a English-like language, right? They also have alphabets. Okay, and the DNA, for example, the base pairs, I'm talking about the base pairs. Okay, when you combine the base pairs, they have special meanings. Okay, for example, ATGS, ATGT comes together, they have a special meaning. Okay, so it's similar to a English-like language. So you can use LLM models for biomedical field also. Okay, so what are the different fields we have in bioinformatics? For example, we have protein structure prediction. Okay, we have sequence analysis, we have drug discovery, we have gene expressions, we have pathway analysis. So there are different areas where you can, in all these areas, you can use large language models to solve these problems. Okay, so we, as I told you, I am just discussing only about this problem. Okay, sequence analysis. So what are the language model that can be used in the sequence analysis? You can use DNA BERT, you can use RNA BERT, you can use Gen BERT, TCR BERT. There are many. Okay, only few are given here. Okay, so these are the pre-trained LLM models that can be used for sequence analysis. Okay, so let's so in this session we are just going to discuss some three pre-trained LLM models that are used for DNA sequence classification. So already I told you about the BERT model, right? So we know BERT model has 12 transformer blocks. The basic BERT model has 12 transformer blocks repeating. And when you give a sentence, it is able to generate text. You can generate text, you can classify. Okay, you can use it for many purposes. I told you BERT can be used for many purposes, not only one. It can predict the next word, it can classify the text, it can summarize the text, so it can be used for many purposes. So how you will use one model for many purposes? You have to change the head. So for example, see here you have a head. Okay, so this is the last layer. Can you see this is the last layer? On top of the last layer, you have to add the head. What type of head you are adding? Based on that, it will do the work. Suppose if I add a classification head, then it will do classification. Okay, so there are different types of head that can be added on top of the model. Okay, now this is the architecture of the DNA BERT. So what they did, they took the BERT model, they trained the BERT model with the DNA samples of many species, and they created a new model called as what? DNA BERT. Now this model can understand the language of DNA and RNA. Okay, so look at the architecture diagram here. So here you have the um, DNA sequence. Okay, so you give the DNA sequence. The first step that you're going to do is do tokenization. There are different tokenization methods available. Okay, to convert this into tokens. Okay, so we will be discussing shortly. DNA BERT will be mainly using k-mers. Okay, so what is k-mers? I already told you what is k-mers, right? Okay, so here they are using 3 k-mers. Okay, for example, take the first 3, then the next 3. See, first 3 is AGC, the next 3 is GCA, the next 3 is CAC. So it takes overlapping k-mers, right? Overlapping, and the K is 3. So this is a token. So when you give your DNA sequence as input, it first convert that into tokens, and then it adds some special tokens. Okay, the CLS and SCP are special tokens. So this is for classification and this is for separation. Okay, so it separates one DNA sample from another DNA sample by adding this tokens. So special tokens are added. Then you mask some tokens. See, I want my bird to understand my data, right? Whatever data that I give, it wants to understand the data. So to understand the data efficiently, what it will do, it will mask some token and try to predict the mask tokens. Okay, so that is how it is getting trained. The bird is getting trained by masking some tokens. Okay, so you can change how many tokens has to be masked. Okay, all those things you can change. Now that is given as an input to the token embedding. So what will happen? Each token will be converted into a vector. To that vector, you are adding the position. To that position, that is what is given as an input to the transformer. Okay, now it passes through multi-head self-attention network, then the normalization is added, then feed-forward network is added, then normalization is added. So normalization layer are actually added to reduce the complexity, because the vector will be very huge size. To reduce the complexity, dimensionality of the vector, you add normalization layer. Okay, so and the output of this one will be what? Vectors. The output of this one will be vectors. Okay, so how many tokens here you have? That many tokens. See, for each token you will have a vector. For each token you will have a vector. And what will be the size of the vector? The size of the vector will be number of neurons in the hidden layer. Okay, so we saw in hidden layer, I think we have around 768 units, neurons are there in the hidden layer. So for each token you will have a vector of size 768. Okay, then what you want to do with your bird? Do you want to do classification? Then you will add a classification head here. Okay, or you want to do something else, then you will change the head. So you will change the head based on what you want to do with your BERT model. Okay, so in other words, I can tell your BERT model can be you can down sample for any task that you need. Okay, you can predict the next sequence, or you can classify the sequence. Okay, like that you can use it for different purposes. Okay, or identify the binding. Suppose you want to identify a binding in the DNA, that also you can do. So like that you can use it for different purposes. Okay, so this is your DNA BERT architecture. Now this is a Python code how you will load the model. See, it's called as sometimes you directly give the DNA sequence. It will predict because it's already trained on the DNA samples. Okay, the model is already trained on the DNA samples. So without doing any training, your model will be able to predict the class. So that is called as zero-shot learning. Okay, so without learning your data set, the model will be able to classify because it's already trained on the DNA samples. So how to load these models? Just you have to load. It is there in the Hugging Face library. So what you have to do, you have to just download the model. So here you can see you have to give the name of the model. See, in BERT you have different models, like you have DNA BERT 2, which uses 2 k-mers. Okay, then we have DNA BERT 3, which uses 3 k-mers. DNA BERT 4, which uses 4 k-mers, 5 and 6. You have different, okay, so you can repeat your algorithm with the different options and check which one is the better. So maybe your data set will be better for 2, 3 or 4. So you have to do a trial and error method. So you have to load the model, and for each model you have a tokenizer. Okay, and this is the configuration information about the DNA BERT. Okay, so now we give the sequence. So this is your sequence. See, this is your DNA sequence. Actually, I have taken a very small length. Mostly the size will be around 20,000. As you know, if you take any viruses, it will be sometimes around 26 base pairs, 26,000 base pairs. Okay, so the important difficulty that you face will be the memory. Your memory capacity will not support to run such a long sequence. So I have cut the sequence. The sequence length is only 100 here. Okay, so this is your DNA sequence. And now you can see you will use this tokenizer. So you will use this tokenizer and you will pass this sequence. And you can tell you have to add the special tokens. We saw there are two special tokens, right? One is for classification, other one is for separation. And then you can add whether the token has to be masked or not, right? So you can see when you give your DNA sequence as input, you can see the DNA sequence is converted into the tokens. Can you see here? It is converted into tokens. So here it is using, this is your tokens. Now you give this token as an input. See, you are giving this token as an input to your model. Your model, model is nothing but your transformer. So you're giving that as an input to your transformer, and then directly you can take the output of the last layer. Last layer is your hidden layer. So can you see here output, hidden layer? Okay, I'm taking the vector of the first token. What is the first token? First token is CLS, right? That is your first token. So your first token is converted into a vector. Can you see? It's a long vector. Okay, the size of the vector is around 768 because you have 768 neurons in the hidden layers. So this is the last layer output. Now you can take the output of this last layer and you can also feed this one into a machine learning model and classify, and you can classify. So one thing what you need numbers, right? You need numbers. You have to convert your DNA sequence into numbers. So when you take the output from the last layer will be the numbers, okay? The vectors, okay? You can just pass that into machine learning model and or some deep learning model like CNN, one-dimensional CNN, or to an LSTM, and then you can classify. Or you can continue with the natural language processing. So this is your last hidden layer. On top of this hidden layer, I will add a head. What head I want? I want to add a classification head. Okay, I will add a classification head. So when you add a classification head, you will have a softmax layer. Softmax layer will convert this vector into probability. Okay, so which word is which token is having the highest probability? That will be your next token. So that is how the training happens in the BERT model. Okay, so this is the first pre-trained model which was created from the BERT, and they renamed it as DNA BERT. So there are different versions, like you have DNA BERT 2, 3, 4, 5, and 6. So you can repeat your experiment with different versions. Okay. Okay, the next model that we are going to discuss is GPT. Okay, the GPT model, which we are frequently hearing every day, GPT, right? So GPT model is customized on DNA samples, and it is called as DNA GPT. Okay, so this model is inspired from GPT. What is the full form of GPT? Generative pre-trained model. Okay, so here it is trained with multiple DNA sequences from various species. Okay, various species. Okay, on what all species it is trained? The species names are given here. Okay, so on all these species, the DNA samples are taken, and the DNA GPT is trained on this one. So it's trained on 10 billion base pairs from 9 different species. So if your DNA sample is very long, see, one important thing I forgot to tell you, DNA BERT will work only for sequence of length 512 base pairs. Okay, the example here I showed you the DNA sequence of 100 base pairs. Okay, the base pairs, you have only 100 base pairs here. DNA BERT will work for 512 base pairs. More than that, it cannot process. Okay, but if you have a very long DNA sequence, you can use DNA GPT to process it. Okay, so it has a capability to process a long DNA sequence. Okay, so this is the architecture diagram of DNA GPT. So here, can you see the speciality about this model is you can give DNA as an input, or you can also give just token numbers as input. So it can accept both as input, the sequence as input or numerical sequence as input. So both can be processed by the DNA GPT. Okay, and these are the layers you have. You have masked multi self attention layer. Okay, self attention layer. Then you have normalization layer, feed forward layer, then normalization layer. So this is the basic architecture. So this architecture is similar to your transformer, right? And how many repetition you have? You have 12 transformer layers here. Okay. And what will be the last layer? You have to add the head. So here you can add the classification head if you want to do the classification, or you can add a regression head if you want the numbers. Okay, if you want the output in numbers, then you can add the regression head. So based on what head you are adding to the model, that work you can do. Okay, so this is the DNA GPT model. Okay, so this is how DNA GPT will work. So here also how it is divided into tokens. It is using k-mers to divide into tokens. So the sequence are divided into k-mers. K-mers are converted into numbers by passing it to embedding layers. So either you can pass a k-mer and convert that into numbers, or directly you can pass the number. Okay, if you have the number also, you can directly pass a number to the numeric embedding layer. So that is specialty about DNA. So it accepts sequence as well as it accepts tokens as input. Okay, and both are sent to the model, and the model will process, and it combines the generates the output, and the output is given to the head. So there are two heads are available in this model. Either you can use classification head or you can use the regression head. Okay, so this is how DNA GPT will work. Okay, in this DNA GPT, the tokens that it is using, okay, it treats the DNA sequence in a very special way. Okay, so here in DNA GPT, you can see in this architecture, it uses multi-self attention okay, mechanism. So it tries to understand the meaning of the DNA sequence. Okay, so when, for example, how a regular language is, right? Each word has a meaning. Like that in DNA also, when certain base pairs are coming together, there is a meaning for that one. Okay, so what this will do is it will generate k-mers, either non-overlapping k-mers or overlapping k-mers, and form DNA words that are meaningful. And DNA words will be combined together to form DNA sentence, and DNA sentence will be combined to form DNA paragraphs. So what is important here? It understands the base pairs. Okay, only certain base pairs will come together. Okay, for example, I can tell always, for example, let's take this sequence. Yeah, see, for example, GA. Okay, so this will come together always. Okay, for example, TGGA, this will come together always. So there is a special meaning for this one. There is a special meaning for this one. So DNA GPT, it understands the DNA language. Okay, so it combines the individual base pairs to form a DNA word, and DNA words are combined to form DNA sentence, and DNA sentence are combined to form DNA paragraph. Okay, so this is the importance about DNA GPT. Okay, so it follows a brilliant architecture. Okay, so the tokenization method that it uses, okay, that is the difference in DNA GPT. The DNA GPT is better. Why? Because it uses a brilliant architecture to understand the DNA language. Okay, so it has special tokens like instruction token. Okay, so whenever you want to predict the next token, you can give instruction token. When you want to tell some tokens will be connected together. For example, I told you, for example, here I told you, whenever you have GAGTAA, the next token will be always CCAGA. Okay, certain tokens will not join together. Okay, so this one you can tell whenever this token comes, the next token will be always this one. So I can tell a connection between these two tokens. Okay, so I can represent the connection tokens. Okay, then I can also assume classification tokens, regression tokens, numbers, and also some reserve tokens. So the tokenization method that is used by DNA GPT is little different from the BERT model. Okay, it is different from the BERT model. Okay, so this diagram here you can see, this is the sequence. Okay, so this is a sequence here. So either you pass a sequence or you pass the numbers. This is nothing but the token numbers, okay, as an input to your DNA GPT. And then it has special tokens to indicate which one will together. For example, if there is a relationship, it will put a plus. Okay, if both are equal, it will put equal to. So like special symbols are used, special symbolic languages are used to denote the tokens. Okay, so in your DNA GPT, and there are different versions are available for DNA GPT. For example, you have DNA GPT H, DNA GPT M, DNA GPT S 512, DNA GPT B 512. So different versions of DNA GPT models are available, and you can see the number of parameters that are tuned. You have 0.1 billion parameters, and the sequence length. See how long the sequence length. DNA BERT is only 512 base pairs. And see here, it is around 24,576 base pairs it can process. Okay, and how this is already pre-trained? It is pre-trained on human genomes. Okay, genomes. So if you want to predict the human genomes, then you can use DNA GPT. And this one is trained on 9 different species. This is for all mammals. Okay, so based on your data set, you can select which version of DNA GPT you want to use for your data set. Okay, sometimes see, mostly you will be using this model as it is available. Okay, you have very less work. You just have to download that model from Hugging Face, and then just you're going to add the head alone, and then you're going to train on your data set. Okay, so it's called as zero shot learning. Without learning also, sometimes it will be able to classify. Okay, if the accuracy is not better, then you can start training the data set again. Okay, so I will show you in the code how it is retrained on the data set. The last language model that I'm going to discuss for today is called as GENA-LM. Okay, so it is a open source foundation model, and it is mainly used for long DNA sequences. Okay, so this is also masked language model only. So most of the language model we discussed is masked language model. What is this masked language model? How these models are learning your data set by masking the few tokens, and it will try to predict the value for the token. Like that only this model learns your data set. That is why it is called as masked language models. So these model is also trained on human DNA sequence. So this one and your DNA GPT are trained on the human DNA sequences. Okay, so but this GENA-LM model, it uses a different tokenization method, which is called as BPE, byte per encoding tokenization method. It is not using the k-mers. Okay, and the maximum length of sequence it can process is about 4.5 thousand to 36,000 base pairs when compared to 512 base pairs in DNA BERT. And you also have another model called as nucleodic transformer. That is also a very good model that can able to process 1,000 base pairs. Okay, so so if you have a very long sequence in your data set, you have very long sequence, then you can use GENA-LM. But I told you there are some we have difficulty in training those models. If you have a very large memory capacity, then you can use long DNA sequences. Okay, so now I just want to tell you what is this byte per tokenization. It is completely different when compared to k-mers. So k-mers, we know it will take either 2 or 3 or 4 overlapping or non-overlapping k-mers, but byte per tokenization is completely different. So here, for example, this is your input data set. Okay, this is your input data set. From the input data set, it will create a corpus. Okay, it will create a corpus. So what will be the corpus? The corpus will be the unique letters from all these data sets. So what are the unique letters? You have A, B, C, D, E. Okay, so this will be your initial corpus. It will start slowly creating corpus. Okay, the corpus will be keep on adding. So how it will do? This is your starting corpus. Now from this, it will start creating the frequency. For example, take the first word. How many times A appears? This is your given input, right? So A appears only once. B appears, you can see 3 times. Okay, C appears 1, 2, 3 times. So it calculates a frequency. Okay, the next step, it takes the highest frequency pair, 2 characters of highest frequency. So which one is having higher frequency? B and C. Okay, and it combines. Okay, it combines, and then it calculates the frequency of B and C. So from this one, so BC, you can see it appears 2 times. It calculates the frequency, and then it adds to this vocabulary. Okay, so it adds to this vocabulary. See, for example, like this. Okay, so now a new word is added to the vocabulary. So how it is added? It just finds out which is the most frequently occurring word, and then it combines and creates a pair. Okay, and then it merges into the vocabulary. Now the steps, again, these steps will be repeated. Okay, the same steps will be repeated. Then it forms 3 pairs, then it forms 4 pairs. Okay, like that it keeps on repeating, and only it will be added if its frequency is high, otherwise it will not be added. So that's how it creates the entire vocabulary. So after creating entire vocabulary, now when I give this text, it will divide this into tokens. See, for example, this is a now which is your vocabulary, this is your vocabulary, right? So A is a separate word, B is a separate word, C is a separate word. BC will be divided as 1 token. Okay, will be treated as 1 token. So wherever BC comes, see, BC will be created as 1 token, and D will be a separate token. DC will be as a separate token. Okay, so did you understand? So these steps will be keep on repeating, and then it will start populating the vocabulary. So until the vocabulary is of some specific size. So you have to tell stop this process when my vocabulary size reaches to 10,000 words. So like that, you have to specify when you want to stop this process. So you have to repeat this process continuously until you have a predefined size for your vocabulary, and then you have to stop this process, and then you give your input. Your input will be divided into tokens based on the vocabulary that is created. Okay, so this is how tokenization will happen in the GENA-LM model. Okay, and this is a Python code to download the GENA-LM. So here you can see you have to give the name of the model. Okay, there are different versions of GENA-LM model. One is GENA-LM bird-based model. Another one you have big bird model. So you can work with both and check which one is the better. Okay, so see, just I have shown this one to show you how to add the head. Okay, see, this is your model. Okay, you have to download the model from here. Okay, and see, you can see the class of this model. See, you can print the class of the model. You can see what are all the classes. It means there are different classes here. See, bird for sequence classification, bird for multiple choice, bird for token classification, bird for next sentence prediction, bird for mask language model. Okay, bird for pre-training, bird for question answering. So you can use this model for many tasks. Okay, downstreaming task. You can use it for many downstreaming tasks. Which task you're going to use based on what head you select. See, I'm selecting the head here. See, TLS. I'm selecting the head class label. I'm selecting the head. What head we have selected? Bird for sequence classification. So one model only, but it can be used for many purposes. Okay, for what purpose you're using your model, it depends upon what head you add to your model. Okay, so these are all the classes you can see. So for any model, you can check the classes like this, and then you can select what head you want to add on the top layer. Okay, so as I told you, GENA-LM is coming in different versions, right? One is using bird-based model, other one is using big bird-based model. So you can use both and check which one is the best. So big bird for sequence classification. So this is your head now. Okay. Okay, so downstreaming task, as I told you, you can use it for many different purposes. So you can go through all these. These are all tool names. Splice AI, Deep Star, Deep Sea, Apparent. Okay, so these are all different tools. They are all using GENA-LM for different purposes. Okay, so you can go through and check. So at last, so we come to an end. We finished discussing all the language models. So there, as I told you, there are many language models. I just discussed only 3. One is DNA BERT. Then we discussed DNA GPT. Then we discussed GENA-LM. Okay, 3 models only we discussed. And mainly, I discussed only how the model is used for DNA sequence classification, right? So you can use these models and do many things. For example, you can use these LLM models for biomedical text mining. Okay, so for example, you can see here you have LavaMed. So this is actually developed by Microsoft, and they They are trained on PubMed. You would have heard about PubMed. They actually publish biological related uh, papers. Okay, so PubMed, they are trained on PubMed, so they can this model actually it creates conversations using pictures and text. Okay, like a GPT model. Okay, the next one you can see PubMed BERT. This is also this is also developed by Microsoft only. So this also is specialized in biomedical text analysis. So here also you can use your pre-trained models. And BioBERT, okay, this is developed by Korean University. It's mainly used for NLP task. It's used for named entity recognition. So maybe, for example, when you give a medicine name, it will able to recognize, and it may tell you for what this medicine is used for. If you tell a disease name, it will able to recognize a disease. Okay, so named entity recognition, and it also answers questions. Okay, BioBERT. MedPalm, okay, so this is created by Google, and this also answers biomedical questions. Okay, PubMed GPT, okay, so this is developed by Stanford, and it is specialized in biomedical text mining. Okay, then you have BioLink Bed. So here you can see there are a lot of models are created like Chat GPT. Lot of models are created with these pre-trained models, and each one is used for different downstreaming tasks. Like some are used for classification, some are used for question answering, some are used for text mining. Okay, just change the head, that's it. So you can customize these pre-trained models and use it for your one purpose. Okay, so this is for text mining. For drug development also, you have some models like drug GPT. Okay, so this one simplifies the drug design process. Okay, so it also increases the speed and accuracy of the process. So it can be used to create new drugs. Cancer GPT. Okay, this is developed by researchers in Texas University, and it also predicts the outcome of drug combinations on tissues of cancer patients without requiring extensive data or features. So like this, you have different models. I just named only a few, but there are different models which are customized for each use. Okay, so with this, I finished my presentation. So before going to the question session, just I want to show you the codes. So can you see my picture here? Can you see my screen?
Yes, ma'am.
Yeah, so here if you see, I just implemented one or two models. So first we have to install the packages, the transformer packages, okay, PyTorch. Okay, then you see I have downloaded the models. Okay, the first one I have used is DNA BERT 2. I told you have 2, 3, 4, 5, and 6. Okay, so just we need the tokenizer, we need the model, and we need the configuration. Okay, so we download the model, and now I'm printing the model. See, this is your model. See the model here. Can you see the top layer? The top layer, this is your last layer, the hidden layer. Can you see here? The output feature will be 768. Okay, the number of neurons you have is 768 neurons. On top of this layer, you have to add the head. So the head that we added is classifier. Okay, so if can you change the head? Yes, you can change the head. Okay, so now this is a sequence we are giving as an input, and we are calling the tokenizer. So when you give the input, automatically the sequence is converted into tokens. Okay, and the token is given as an input to the model. Okay, and you can get the output from the hidden layer. See, this is each token will be converted into, see, I told you it is a big vector of size 768. So this is for one token. Okay, this is the last layer. This is for one token. So you can take this output and give it to a deep learning neural network or any other thing, or you can continue to add a head and then do the classification using NLP task. Okay, so this is for one sequence, right? So now we want to do for different. So I have a data set here. See, actually I took the DNA samples from three. One is coronavirus, other one is MERS, and then SARS. So I took these three viruses DNA samples from NCBI and I combined and put into one data set. So this is my data set, and you can see the number of rows are around 14,988 rows I have. So I have the sequence here, I have the sequence length here, okay, and I divided my data set into training and testing. And here you can see the training data set. See, this is your sample, and I'm restricting my sample length to only 100. As I told you, I have some memory issues. So I'm running my thing in GP only, but still I face some memory issues. So I'm not able to run for a very long sequence, and it will take very long time to get the output also. Okay, so now this one, yeah, this is the KMERS. Okay, so now, so what I do, the entire data set I convert into tokens. Okay, and then I add some padded tokens. So why I want to add padded tokens? Because each sequence may be in different length. Sometimes I need to add the padded token so that I make all the tokens of same length. Okay, so now this is my tokenized data set. So it has two things. One is training, other one is testing. Okay, now I train the model. Okay, so this is to train the model. So here you can see the models are trained. The training happens. I just run for a very short epochs, 10 epochs. See, I'm able to generate a good accuracy of around 92. Okay, so for example, you can see, yeah, it's around 93. Okay, so this is about one model, and this is the AOC curve for that model, and this is about GENA-LM. So I told you in GENA-LM also you have two versions. One you have Big BERT, other one is BERT base model. Okay, you can use any of that you wish. Okay, so here you can see the last layer. The last layer is a classifier layer. So you are adding the classification head as a last layer and then running to the same data set. So here the tokens will be a little different when compared to the DNA BERT. Okay, the tokenization will be different in this one. Okay, and then training the model. Okay, and got around here also you got around 93%. Okay, and this is your last model, GPT model. Okay, so this is the GPT model. So here I did not add the last layer. Actually, I added here. So here, if you see, I run this model for yeah, I created a stratified fold. I created 10 folds. Okay, and then I run this model. This is DNA GPT. And I got the maximum accuracy of around, I think, 94 for DNA GPT. Yeah, 94, which is greater than the other two models. 94. I run for 10 folds. The previous one I showed you, I just run only once. This is, yeah, 95. 95. Okay, so when I run for 10 folds, I get a good accuracy. So here you can see the nature language are much better than the traditional models. When you take machine learning, they got around some 93. Deep learning CNN models, they got around again 93 or something. But this natural language models are much better than the traditional models. So they are able to understand the DNA language much better when compared to the other traditional models. Okay, that's all is my presentation. Any questions?
Yes, hello, ma'am.
Yes, sir.
Yeah. Actually, I'm not an expert. Actually, I'm a mechanical engineer. So first
Yeah.
of all, I would like to congratulate for a very, very good lecture. I mean, it was a thorough lecture. And I just have some basic question,
Yeah, tell me,
You
sir.
talked about, when you talked about the transformer model,
Yeah.
And you talked about encoders and decoders.
Yeah.
What is the functional, what is the meaning of the word encoder and decoder when you talk of these models, large LLM models?
Encoder is trying to
Mhm.
convert from one representation to other representation, to encode the information. For example,
Mhm.
you want to translate from one language to another language.
Okay.
Okay, so encoding is a process of trying to understand that language, and decoder will convert that language into another language. So encoder does one job, decoder does another job.
Okay. Okay. And you also in your initial part of your presentation, you talked about RNN, LSTM, and then
Yeah.
The thing moved to the large language models. So,
Yeah.
can you tell me in brief what is the basic difference between these two? Because nowadays we are talking a lot about this LLM
Yeah.
based on transformer. I mean, just in a single, can you tell me what is the
Yeah,
basic
that's
difference?
what, see, sir, that's what the LSTM and RNN, they are able to understand sequence. Okay, so for
Okay.
example, I want to predict the next word.
Mm-hmm.
Hi, if I tell hi, what will be the next word? So it predicts the next word. How are you? Okay,
Okay.
but this LSTM and RNN,
Mm-hmm.
LSTM will predict the word, but just seeing the before word. Okay,
Okay.
and it can see the last few words.
Okay. Mm-hmm.
Okay, so LSTM has a limited capacity, so it can see only the last few words. But RNN, if you take, it can see last some few words more than the LSTM. So they are
Okay.
all they are all used for sequencing, predicting sequence, but
Okay.
they cannot see bidirectional,
Mm-hmm.
and they cannot understand the context. So if you want to
Okay.
understand the context and do bidirectional, then you move to the transformer.
Okay. Okay. So these were just, they were just invented in 2018, you said.
Yeah, yeah.
Okay. So now
2016.
all the LLMs now which are being used, they are based on this transformer model.
All the LLMs, yeah, they are mainly based on, that is a base.
Okay.
Transformer is the base. And they
Okay.
add
Base.
some few on top of it and they create a new model.
Okay. 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 Thank you, ma'am. Thank you.
Thank you, sir. Any other questions? Okay, one of them has raised the hands.
Dear participants, now you can raise your questions for clarifying your doubts. So thank you, ma'am.
Thank you,
Thank
ma'am.
you very
Thank you
much,
very
ma'am.
much.
It is a really a very informative section, ma'am. You have clearly and detailedly explained all those things. Thank you, ma'am. Dear participants, I have posted the feedback link in chat box. Kindly fill it. Thank you very much, ma'am. And thank you all.
Thank you for the patient listening. Thank you very much, dear participants. Thank you.
Thank you.
Thank you, ma'am.